Hello, everyone, and welcome to Bio 181. In this lecture, we will be covering Chapter 1, The Study of Life. More specifically, we're going to take a look at what the science of biology is. And to understand that, we need to understand the nature of science and the nature of biology. Let's begin with the question of what is science. We may find a variety of different definitions on the internet, and they all seem to point in the direction that science is a process to understand the universe. However, none of them describe in detail what is the process in which this is accomplished. Additionally, as we'll discuss later, the outcome of, quote, knowing extends beyond the scope of science. Science uses the same methods we use on a daily basis of observation and reasoning to better understand the natural world. However, it does this with more stringent rules with the goal of collecting evidence to justify explanations. It uses a method of proposing and testing explanations to gather evidence to lend support to conclusions. As a warning, science shares homonyms, or the same word that we use in day to day living, but with specific and distinct meanings, and we'll need to take time to distinguish these to understand the process of science. These scientific explanations are the basis of hypotheses, which explain natural phenomenon or a set of observations. Hypotheses can then be used to make predictions of outcomes for further tests, and by doing so, become validated. A hypothesis must be demonstrated to be accepted. Remember that it is the testing of a hypothesis that validates it. If one cannot fathom a test, which could potentially falsify the hypothesis, then the claims and explanations made are not within the realm of science. For this reason, science must always keep open the door for revision. In this way, science cannot claim to know something or prove a claim as absolute truth. Rather, its goal is to better approximate our understanding of the universe. Look at the following two explanations. Explanation 1 is an explanation that could be tested and give a result that supports the claim or not, and this is what we mean by it being falsifiable. Explanation 2, although to most of us clearly seems false, cannot be tested and therefore cannot be falsified, so it will not fall under the realm of science. Some people will say that a scientific theory is just a theory and not to be taken seriously. But as we discussed earlier, some scientific words have distinct meanings compared to how we would typically use them in our own conversations. A scientific theory is one of those words. It would be perfectly natural for us to use the same word theory in a way that is more similar to an untested hypothesis. For example, I have a theory that because I didn't talk to my Uber driver, he charged me extra for the ride. However, a theory is a hypothesis that has stood the test of time. It has been so rigorously tested and verified through many different scientists' independent inquiries and angles that it has become broadly accepted as a model for understanding and having strong predictive power. Here are some examples of well known theories. Take a moment to wonder, how do scientists obtain such theories? As you can imagine, these theories are not just some ideas or one off experiment. These are the culmination of a vast collection of scientific papers, data, and analysis, and through it all have been incredibly resilient to testing and useful in modeling and predicting outcomes to experiments. The scientific method is often presented in a linear fashion, from asking a key question to doing background research, constructing a hypothesis, testing the hypothesis, analyzing the resulting data, and reporting. However, keep in mind that science usually takes a circuitous path where there may be several competing hypotheses and a series of experiments and analysis in tandem or series, and even revisiting or refining hypotheses along the way. Let's now take a look at the components of every experiment. Experiments involve variables. Variables are things that can be altered, either by the scientist or experimental design directly or indirectly. The two types are independent and dependent variables. Independent variables are those that may be directly altered, while the dependent variable is altered indirectly by how the dependent variable is set up. The following is an example of how this works. Let's suppose I'm trying to determine whether feeding my chickens more sunflower seeds will cause them to produce more eggs. Now recall that a variable is a component of the experiment that can be altered, and dependent variables can be altered directly, and independent variables are the consequence of dependent variables. In other words, they're altered indirectly. So can you identify the dependent variable in this experiment and then identify the independent variable? If you said that the independent variable is the sunflower seeds in the diet, you would be correct. That's something that we can directly adjust for the chicken. If you said that the dependent variable is the number of eggs produced, you would also be correct because this is dependent on how the independent variable was set up. Oh, here is an experimental design of the previous example. We have the two variables of providing seeds in varying amounts and the amount of eggs. However, can we reliably show that there is or is not a relationship between the two? The answer is no, in part because there were multiple factors involved, such as when the feedings took place, when the eggs were collected, which could have meant more time to lay eggs, and lastly, the number of days on the diet. A good experiment must only vary one variable at a time. Otherwise, another variable may interfere with the results. And that brings us to our first checkpoint of the course, which is you are trying to determine whether drinking more coffee improves performance on Bio 100 exams. What are the independent and dependent variables in this experiment? And if you need to, go ahead and pause the recording to write down the question. Checkpoint two is you are trying to determine whether drinking more coffee improves your performance on Bio 100 exams. What are some variables that you should control? In other words, keep consistent over the varying experiments in order to ensure that your results are valid. Now that we have covered variables, how can we use them to support or reject conclusions? Here we have assumed all other variables are controlled or kept consistent, and the only independent variable we manipulate is the number of scoops of seeds. We can see that there is a positive correlation to the scoops of seeds and eggs laid. This type of reasoning is called inductive reasoning, where we have used specific data to develop general conclusions. And taking this a step further, we have developed a general conclusion that the number of eggs produced is related to the number of scoops of seeds. From that, we might predict that 5 scoops will lead to 7 eggs. And this is an example of deductive reasoning, or using general conclusions to make specific predictions. To bring it all together, specific experimental data allows inductive reasoning to create general conclusions, which, through deductive reasoning, can be used to make specific predictions. Checkpoint 3. 400 years ago, Galileo used his observations of the solar system to develop the theory of heliocentricity, which states that the sun is the center of the solar system. In 2017, astronomers used that theory to predict the exact date, time, and location of the total solar eclipse. Each of these instances represents what type of reasoning? Checkpoint 4. The theory of natural selection says that organisms that are more well suited to their environment will survive to produce more offspring. On the basis of this theory, you predict that giraffe B will survive to produce more offspring than giraffe A. This is an example of what type of scientific reasoning. Scientists need ways of ensuring the design of the experiment is sound. To do this, they will purposely set up parameters to yield a positive result and another experiment to yield a negative result. These are called the positive control and the negative control. For example, you might feel like you are sick with a fever, but you're not sure if your thermometer is working properly. In a sense, this is an experiment, and you may want to design controls to see if the experiment is working properly. If you know that your brother, for a fact, is currently sick with a fever and your sister is perfectly healthy, you could design a positive and negative control around this. What are you testing for? Presence of a fever. And in which individual do you expect a positive result? Well, the brother who has the fever. And which individual do you expect a negative result? Sister. And what would it mean if the thermometer reported a normal temperature in your brother? What would it mean if the thermometer reported a fever in your sister? Checkpoint 5. If a swimming pool is acidic, it is generally not safe to swim in. You plan to use a pH strip to test the acid level in your pool and determine if it is safe. Describe how you would set up a positive and negative control for the experiment to determine whether your pH strips are working properly. Sample size describes the number of times that a particular test is carried out. The greater the size, the more confidence we can have in the outcomes. Following this checklist will increase your odds of a successful experiment. Testing only one variable at a time, using valid scientific reasoning, whether inductive or deductive, applying experimental controls, and having a large sample size. Checkpoint 6. Describe the distinction between a hypothesis and a theory. Okay, now let's discuss a little bit then about what the nature of biology is. Um, Biology is the study of living organisms. And while that seems rather intuitive for us, what exactly is included within the definition of living organisms? Taking a closer look at this question, there are going to be 8 requirements for something to be categorized as a living organism. It has to have sensitivity or response to stimuli, be capable of reproduction, adaptation, growth and development, regulation, homeostasis, energy processing, and order. The first item on the list was sensitivity or response to stimuli. Organisms must be able to respond to signals from their environment. Examples listed here are jerking your hand away from a hot pan, or sunflowers tracking the movement of the sun. This is called phototrophy. Or microscopic organisms moving towards a food source. This is called chemotrophy. Finally, living organisms need to have order. They are made of molecules that are the building blocks of cells. Multicellular organisms need to be able to organize into tissues, organs, organ systems, and bodies. This slide has a bottom up approach of biological order in the physical realm. We have the macromolecules, which are made up of the 4 building blocks: carbohydrates, lipids, proteins, and nucleic acids. We have organelles, which are subcellular structures that serve a function within a single cell. Then we have the cell, and these cells make up tissues. Tissues then make up organs, and organs then make up organ systems. Point 7. During the Voyager missions to Mars, an experiment was conducted on samples of soil that shows evidence of metabolism. Did the Voyager missions prove that there are soil microbes living on Mars? With recent advents in genetic engineering, research, and biotechnology, the applications of biology are beginning to expand quite a bit here. For example, in 2017, we have the first gene therapy approved by the FDA. This particular gene therapy was to treat a form of retinal disease. Additionally, we had FDA approve the first gene therapy for leukemia. And we also now have the Arctic apple slices, which have the gene responsible for browning in apples removed. Now can make silk fibers in the lab, and people can clone their pets. And now we have ways to engineer what they call gene drives, which are these genes that lead to essentially destruction of the offspring that are being applied in mosquitoes. These are already in the testing phase out in the field. We're being able to do genetic testing on remains to discover new groups of populations in our past. And we are also working at analyzing the base chassis, metabolic chassis of a cell. So this synthetic microbe lives with fewer than 500 genes. And to put that into context, a human has about 25,000 genes, and an E. coli has about 4,500 genes. So scientists are trying to research and understand what are the essential components in order to operate a cell. Anyway, that concludes chapter 1. Thank you so much for joining, and we'll see you for the next chapter.